I've been patiently waiting for this track to explode on. Welcome to this deep dive on Mots-C, one of the most exciting mitochondrial-derived peptides in current research. This small molecule is showing remarkable potential in metabolism, exercise physiology, and healthy aging. Here's a clear positive overview of the key findings. Mots is a 16 amino acid peptide encoded within the mitochondrial 12S rNAR gene. It functions as a retrograde signaling molecule. During exercise or metabolic stress, Mots translocates from the mitochondria to the nucleus, where it modulates gene expression to optimize energy homeostasis, enhance cellular protection, and improve systemic metabolic resilience. In healthy humans, a single moderate-intensity 40-minute cycling session elevates skeletal muscle Mots mRNA approximately 12-fold and circulating peptide levels by about 1.6-fold, with these increases persisting for several hours afterward. This strong induction establishes Mots-C as a genuine exercise-responsive exerkine, helping explain many of the broad metabolic benefits associated with regular physical activity. In high-fat diet mouse models, systemic administration of synthetic Mots prevents obesity almost completely. Treated animals exhibit minimal weight gain, significantly improved insulin sensitivity, reduced fasting insulin, and normalized glucose tolerance. These effects are driven primarily in skeletal muscle via robust activation of AMPK, upregulation of PGC1-alpha, promotion of mitochondrial biogenesis, enhanced fatty acid oxidation, and superior metabolic flexibility, the efficient switching between carbohydrate and lipid fuels. Mots doesn't politely ask fat cells to leave. It activates AMPK and hands them a one-way ticket to the bottomless pit. Aging studies provide some of the most compelling results. Endogenous Mots levels decline progressively with age in both humans and rodents. Intermittent dosing in very old mice, late-life treatment, equivalent to human 70s, leads to substantial improvements in physical function, increased grip strength, more stable gait, faster walking speed, and higher overall performance scores. Endurance capacity improves dramatically. Treated elderly mice run considerably longer distances and times to exhaustion compared with age-matched controls, with certain studies showing performance approaching that of untreated middle-aged animals. Cellular protection is robust. Pre-treatment of myotubes with Mots roughly doubles survival under glucose restriction or oxidative stress, sustains higher oxygen consumption rates, preserves efficient oxidative phosphorylation, and maintains stable ATP production. It achieves this by activating NRF2 to boost antioxidant defenses and inhibiting NF-kappa B to reduce chronic inflammation. Mots also mimics several key benefits of calorie restriction without dietary changes. Enhanced ACTI-mediated insulin signaling, PGC1 alpha-driven mitochondrial biogenesis, and improved energy homeostasis. It promotes osteoblast proliferation and bone mineralization in cell models, supports glycogen sparing during prolonged exercise, and increases power output. Human observational data shows positive associations. Higher natural circulating Mots levels correlate with greater lean body mass, superior muscle power, better explosive strength, and more favorable metabolic profiles, especially in active older adults. Preclinical studies indicate good tolerability at therapeutic doses, with effects targeted primarily to metabolic and musculoskeletal systems, and no significant adverse signals reported. Recent research continues to strengthen the case. Mots restores mitochondrial respiration in diabetic cardiomyopathy models, attenuates cardiac hypertrophy, protects pancreatic islets from senescence to delay diabetes progression, and exerts broad anti-inflammatory effects across various contexts. In summary, Mots stands out as a highly consistent mitochondrial-derived peptide that mimics exercise benefits, counters obesity and insulin resistance, preserves physical function during aging, and enhances cellular resilience, all from a remarkably compact sequence. I'll catch you on the next one. I have to take my hot girlfriend and my wife to Benihana. Don't shoot a brick.